So today's lecture picks up where we left off last week, which talked about vision in particular. And today we're talking about attention, which is really what you could think of as selecting from different kinds of information, and that will include vision. So when you see a complex room like this, how do you focus in on some information and not other? But it's not just about vision, it's about how we do this with all the information that we're given through all of our senses. Here, this is a famous example of something called inattentional blindness, which has nothing to do with blindness as we understand it. It's basically that sometimes we don't see things if our attention is elsewhere. So what this is a video that you can look on YouTube, but basically what you have to do is you have to count the number of basketball passes that people have when they're just throwing it around. So it's quite cognitively demanding. They're throwing it backwards and forwards. But basically, the gorilla walks on from this side to that side. He does that, and he walks off. About 50% of people do not notice the gorilla, okay? And about half do. So this here is something that is linked to attention. Your eyes would have seen the gorilla at some level, and I'll show some evidence from similar studies that it probably activated things in your visual brain. But basically there is something that selects information from that visual template and focuses on some instead of the other. And that's kind of what attention is doing. So your attention was on these people, so you missed the gorilla in that case. And obviously if there was no people there, just the gorilla, everybody was missing. So what is attention? I'll talk a little bit about this, particularly in terms of vision for action. So the idea is that there are multiple branches of vision. So one that's involved in recognizing objects and one that's involved for acting on the world, so picking things up, for instance. And then I'll talk about this beyond vision, so multisensory aspects of attention, so the body, sounds, and so on, how these kind of enable us to focus in on certain things and to act on them. And then I'll talk about the difference between the left and the right hemispheres in attention, how brain damage can give rise to a symptom called neglect. So we can think of attention as being involved in selection. So in this image here, the idea is that you might be searching for something. It might be, I don't know, a line or something like this. But you can focus in here on different colours, you can focus in on shapes, but also there would be sounds, there would be smells, lots of things would be going on. And basically all of these sources of information will be competing with each other here. And what you're having to do is to prioritise what is it that you're going to focus on. And you might do this according to what your current goals are, that you're searching for a line, or maybe you're searching for your friend, and so on. And essentially what you're doing is you're taking all these sensory inputs and you're kind of dampening down the ones you're not interested in and enhancing the ones you are interested in. And that might mean that you look for all the green objects, for instance, including ones you don't want, such as a green apple, for instance, here. So there's a competition between different sensory objects and a competition between your goals and what's coming in through the different senses. Action also involves selection as well. So whereas in that scene you might be processing all the different colours and sounds and smells simultaneously, our bodies pose a certain selection demands on us, and that's simply because we cannot do this, is that we have to do one thing at a time in general. I mean, with some limitations, obviously our eyes can do one thing and our hands with another, but even so there's a finite range of possibilities that you can do. So in effect, given an infinite range of sensory stimuli, to some extent which are being processed in parallel, you're having to focus down serially, ultimately, into one. What are you going to do with your hands or your body or whatever it is? So in a way, this kind of, going from perception to attention to action, kind of involves a shift from parallel processing, processing multiple things at the same time, multiple colours, objects and so on, to one, being able to do one thing with your hands and so on. But in reverse, the opposite would be happening. The action would be, so your goals, what is it you want to do? You want to reach for an apple. That affects what you attend to, and it affects what you see. So in effect, your goals and what it is that you want to do with your body, or what it is that you want to obtain in the world, acts like a filter on your sensory flow. It's effectively saying, right, I want to reach for this particular object, so all the other objects I'm going to dampen down and I'm not going to pay attention for them. So you've got this kind of this shift from the top down and the bottom up, with the two kind of trying to narrow in on a limited number of things, maybe down to one thing that you act upon. And people often describe attention in terms of a spotlight. The idea behind a spotlight is several things. So one idea is that you are focusing on some things and not others. It's not everywhere. It's in some locations more than others. People talk about this in terms of limited capacity, that there's only so many things you can process at once. And that might be because of the mechanical constraints of your body. It might be because of the limitations on your senses, that there's only so many objects you can perceive and so on. But certainly the spotlight is inherently flexible insofar as this metaphor works. So maybe you've got a narrow or a wide beam. So the kind of example there is that if you're looking at words, you might want to look at a central letter if that's your task, or you might want to look at the whole word. And basically you can kind of zoom in on one or the other and treat one as being kind of in your attentional focus and the other's outside it. Also, this spotlight isn't quite the same as eye movements. So, for instance, I can be looking straight ahead, and I can have an attentional focus here. So, the proverbial looking out the corner of your eyes, what that means is that your eyes are looking straight ahead, but your attention is somewhere else. Okay? You're not really looking out the corner of your eyes. It's an attentional metaphor that you are directing attention elsewhere. But this metaphor of the spotlight shouldn't be taken too literally. So with this, you kind of might imagine, well, there's only one place in which you can put the spotlight, whereas in fact, there's evidence that you can attend to, say, this location on the left and that location on the right without attending to the bit in the middle. So it's as if you could kind of, I suppose, if you carry with the thing, you could split it in things. So it's not a single beam, it's a flexible. But the idea of the spotlight is that you're processing some things at the expense of others. Some things are falling in the shadow, some things in the light. But this is another example where the spotlight kind of breaks down in the sense that it's not different locations that you're looking at, but you're looking at two things which are in the same location. So here you've got a face or a house, and I can tell you to focus on the house, I can tell you to focus on the face. Eventually, if you carry on staring, your attention will just flip backwards and forwards spontaneously between the two. So here, insofar as you think of this as a spotlight, is that it's an object-based spotlight rather than a spatial spotlight, would be another way of thinking about it. But also, think about what your eyes are doing when it's processing this stimulus. In this case, it doesn't matter where you look, you're still seeing the house and the face. Your eyes don't care at all about this. It's just patterns of light on the back of your eyes, whereas your brain can kind of focus in on one or the other. And in effect, what happens is that when you're asked to focus on, say, faces, that basically parts of your visual ventral stream, so this is the part of the brain we talked about last time in object recognition. So the back of the brain here is your early visual cortex, your V1. And then as you go down your temporal lobes, your inferior temporal lobes, you have regions that are specialized for recognizing objects and some degree of specialization for, say, houses and faces and cats and dogs and the sort of thing that we talked about before. But what happens is that if you ask somebody in this stimulus to concentrate on the face, what happens is that the goal here is to concentrate on the face. And in effect, you've got this frontal to parietal to visual kind of network here that basically you could imagine as shining a spotlight on the face area of the brain. And if you measure this in fMRI with your bold response, what you find is that parts of the brain that are specialized for faces, the activity goes up, and for places, it is relatively down. But of course, you could just change the task. And you can say, right, now your task is to concentrate on the houses and ignore the faces. And basically what you find is that you're in effect flipping between what is activated in your visual stream, goes from the face selective areas to the place selective areas, and you're kind of flipping between them. So basically, we can think of as attention as kind of enhancing or amplifying the activity within sensory systems. It is literally increasing the neural responsiveness. But again, here, remember that your eyes are seeing both. It is just the brain's representation of that information. Then the question is, who is controlling the spotlight? So in this particular example, we could say it's top down. It's based on our goals. The experimental whatever says, look for the houses or look for the faces. You're given some kind of context. But in other situations, things might happen from the bottom up. So there might be things that go on in your visual stream that just capture your attention. So the kinds of stimuli that do that would be sudden changes. So for example, if there's a sudden flash of light, for instance, or a bang, what will then happen is that irrespective of what your goals are, this will capture your attention. This selects itself, if you will. It wins the competition, irrespective of what your goals are. So here, in effect, you've got a visual stimulus being selected and then triggering its own action, where a typical action for a flash of light or a bang is you move your eyes to it and you orient your attention and your vision to find out what it is. But it might be other kinds of more complex stimuli. So here, it might be, for instance, you see a threatening stimulus or something you don't like, such as a snake. And again, the idea is that this will kind of pull the spotlight towards it, by which I mean it kind of enhances its own activation. So your representation of snakes become greater than anything else you're looking at, and that sends a signal forward, which alerts you to do something about it, to either discount it, to run away, or whatever it is. And basically, we can think of attention as, although I kind of rather naively put it as kind of a module in the parietal lobes, as we kind of delve deeper, you'll see that that breaks down into lots of other little circuits. But really, attention is kind of, we can think of it as a brain-level property of selecting information at different levels. So you select information at different levels within your visual representations, that some tend to capture attention more because they're sudden, or we assign certain value to them, but also our goals and our actions and so on can do it as well. And basically all these different levels are kind of always competing, and sometimes we select things based on what they look like, sometimes we select things based on what we need, and so on. And this whole system is very dynamic, both bottom up and top down. So although there isn't a kind of a single module that we might want to label attention, there are some mechanisms in the brain that are clearly involved in kind of this selecting, this reducing the amount of input that flows upwards and downwards in the brain. And I'll talk a little bit about how that happens. So first of all, if we go back to this model here, effectively what we have from the visual cortex are two different kind of branches. One that goes this way, which is involved in detecting what objects are, and one that goes this way, which is involved in kind of selection and action, deciding what is your priority and what it is you're going to act on. And this is what's referred to as dorsal and ventral streams, or the what route, which is your ventral stream that tells you what things are. It's a snake, it's a cup. And your where or how route, which tells you where things are. And this is kind of what's illustrated here. So what is what tells you that it's a face, a house, a snake, or whatever. And this dorsal route here is involved in attention and action. Some people call it where, and what that means is that it's kind of this spatial kind of spotlight metaphor of deciding what is your priority, where is it that you're going to look. And some people talk about it as how, so thinking about how you're going to pick it up or how you're going to act on the object. What are you going to do with it? You're going to look at it, you're going to pick it up, and so on. And essentially both of these are correct insofar as this route here contains neurons that contain what we would call maps of space, but also maps of space that link, for instance, vision with your body. So hand-eye coordination, for instance, it links maps of where your body is with maps of where things are on your retina. And I'll talk about both of those. So that's how you kind of find out where things are. It can be where in multiple space, where things are relative to your body, or where things are relative to your eyes. And in effect, what your parietal lobes is doing is trying to link those kinds of information together in order for you to kind of process it further. What it also means is that these two routes are kind of carrying different sources of information. And that one route might give you an answer that differs from the other. And we can look at this both in terms of normal perceptual illusions, but also in terms of brain damage. So one of the things I talked about last week in terms of object recognition or object constancy, which is your what or your ventral stream, is that they have very large receptive fields. And what that means is that the neurons might respond to something like a pen or a cup, but it doesn't care whether the pen or the cup is here, 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 or wherever it is. Whereas your dorsal stream, this root here, doesn't really care whether it's a pen or what it is, but it cares where it is. Where is it in space? So one is more spatial aspects of vision, and one is more shape and other aspects. So the claim here is that in this case, what's happening is that the visual illusion is being driven by this, and that's the sense that these objects are different in length. But your ability to act on it, your hand knows that they're the same length because it's getting more accurate spatial information, and it's discounting the information about the parallel lines. So here, the claim is that your dorsal stream going into the parietal lobes can accurately pick up the object. It doesn't make your hand grip longer than it is, so it's not fooled by the illusion, even though your conscious vision is. And that was kind of the claim made here. Now, the person in the video obviously doesn't have any form of brain damage, but you can see what happens when you do damage these two streams and show that it has different effects on perception versus action. And by perception, I mean kind of consciously stating what it is you see, and so on. So remember from last time that visual agnosia, so Ch9, who couldn't integrate parts into holes and things like this, is caused by damage to the ventral stream here. So this person, for instance, had problems in recognizing birds, couldn't read text properly, couldn't recognize objects presented as line drawings. But the question here is, can you act on the objects that you can't see? And of course, you expect here that you would be still able to do that. And that's what's found. So David Milner was the guy in the video that we've just seen. And he studied a patient who had a visual agnosia, who couldn't do perceptual matching. So what he had to do here, this is the patient DF. And perceptual matching, what the patient was given, is that they were given a slot, and they had to then rotate an object and say whether it's different. So you have to say whether a slot like this is the same as a slot like that. So it's matching two different parts together, but perceptually. And he's all over the place with this. So the correct solution is kind of shown here for the control. But here, instead of just saying whether two things are the same or different, what's happened is that he's given a letter to kind of post, and then he has to post the letter through the slot with his hands. And what you find is that when he's asked to use his hands to kind of reach to the slot and post into it, he can do it accurately. So here he's able to use vision in order to guide his hand, but without his hand, he can't tell you whether two slots are the same orientation or not. He cannot integrate those two pieces of visual information together, but he can integrate vision and motor information. And similarly here, this is like a T-junction that basically the patient will occasionally do it this way. So again, this suggests that actually it's a she, this patient, that the patient kind of knows the orientation of one of these lines, but makes errors at 90 degrees. So she'll try and post the T through it like that. And it suggests that she's treating this object as being two parts rather than as one whole here. So there are some differences there in terms of what can be acted upon, whether it's simple objects or more complex ones. Whereas you kind of get the opposite profile if you were to damage the other root here, and you get a symptom called optic ataxia. So these patients, if you show them, say, images of a pen, they'll say it's a pen. You show them an image of a car, they'll say it's a car. So they're not agnosic. They can recognize visual objects, but they struggle to kind of do visually guided hand movements onto objects. And these are the kind of errors that they make. So here whereas the other patient could post her hand through the slots, These patients here struggle to do that. They make the kind of errors there that they orientate their hand at the wrong angle, or they miss it, in effect. So agnosic patients can't tell what an object is, but they can post their hand or an object through a slot, whereas patients with optic apraxia can recognize objects, but they cannot post their hand or objects through a slot. So you've got these differences here between visually guided action and recognizing objects. And here, this is your dorsal stream there. So this particular symptom here arises from damage going there. The other symptom I'll talk about later on is neglect, where basically patients have problems in detecting stimuli on one side of space. It also means that they tend not to act towards stimuli on one side of space, but I'll talk about this separately. But I just flag it here so you know it's kind of part of this family of disorders that happen if you damage this other visual route. So the dorsal stream is also called the where or the how route, and what it's effectively doing is that it's linking different kinds of spatial maps together. So it's linking together information about where your hand is in space with information about where visual objects are in space. And it's kind of trying to superimpose visual onto, in this case, bodily and motor information. But it also does the same with other senses such as sounds and touch as well. And in effect, we can think of the parietal lobes as containing multiple different maps of space, and that the brain itself contains multiple different maps of space. So the brain doesn't treat space as being a kind of a single entity. So what we have, for instance, is that we've got spatial maps that represent our sensory surfaces. So, for instance, our retina carries information about the pattern of light, but our V1 kind of carries that information as well, even though there's no light in V1, it's just neural signaling. It carries a spatial kind of map of where things are on the eyes. But knowing where something is on the eye isn't helpful. You need to know where something is out there. You're not reaching into your eyes to find objects. And then what we have would be spatial maps that tell us where things are relative to our body. So I know my hand's here, I can see an object there. So you have something that says, treats my hand as in effect the origin and says, right, 30 degrees up there, there is an object. So here, the origin here is my hand, and the object is defined relative to that, as opposed to the object being defined relative to the center of my eyes in that case. And then you would have locations of objects relative to each other. So where, for instance, one object is relative to that, or kind of your spatial maps of space. So where Marks and Spencer's is a relationship to Churchill Square, for instance. That would be a very different kind of spatial map that isn't really sensory based. It's a bit more kind of abstract, or you aren't kind of in the map, if you will. It's kind of what's called allocentric space. So all of these different tasks, such as finding your way around, picking up a pencil, and all these things involve different kinds of maps of space, which are located in different parts of the brain. So V1, your visual area, contains a retinocentric map. The hippocampus contains an allocentric map. That's how you find your way around. Whereas your parietal lobes are very interested in egocentric space, where vision is in relationship to your body, where sounds are in relationship to your body, and so on. And we can illustrate these different maps of space using a phenomenon called the rubber hand illusion. How many people have taken part in experiments? A few of you. Okay, we've run them over the years, but this is an explanation.
A classical illusion that has been published in 1998 by two Americans, Botvinnik and Cohen, two American neuroscientists, is the rubber hand illusion. The rubber hand illusion consists in placing, let's say, one's left arm out of sight, out of vision, and placing in front of the eyes of the subject a hand in plastic, a rubber hand, and then the experimenter will stroke, let's say, the index finger of my left hand, and at the same time the left index finger of the rubber hand. And if he does this synchronously for a minute, up to 2 minutes, certain subjects, 2/3 of all subjects, will start experiencing this hand for some moments as if it were belonging to their body. So this is one way, and this is normally investigated by questionnaires. So the subject will be asked a certain number of questions to respond to. And then there's a second measure, which is, in my opinion, even more interesting, because if you then, after these 2 minutes of stroking, will close the eyes of the subject and ask them, well, now point to where your hand would really be, they do not point with eyes closed to the real arm, they will point towards the rubber arm.
I really felt that my hand was transposed and into that false hand that was presented in front of me. The feeling was really an awkward feeling, a very strange feeling of having a false hand belonging to me. We visually see that the hand is false, but we can't separate that from the fact that we actually feel our hand in that place.
By simultaneously stimulating fake rubber hand and the real hand, something that is felt and something that is seen on a hand is integrated. Under normal conditions, when I see something close to a hand and I feel touched at the same position, this is always on my hand, at least I guess in 99% of all cases.
Okay, so what is happening there is that you can't see your own hand, you can see the dummy hand, but you stroke both at the same time. And here, in effect, you're confusing a visual spatial location, what you're seeing with the dummy hand, with a felt location. So you're mixing up your map of where you think your real hand is in space with your visual information of what you're seeing. And the way that this is measured is, as he said, they're questionnaire measures. You ask people, did you feel as if the dummy hand belonged to you? And people say yes. And another way of measuring it is that you ask people, where is your real hand? And when they're in the illusion, they get confused about where it is, and it feels like your real hand is a few centimeters over from where it really is. So it gravitates towards the visual location. There are other ways of measuring the illusion, and that's you can use something like a skin conductance or startle response. So what happened there is that somebody came in with a fork and stabbed the dummy hand, and he had a very strong kind of reaction to that. You could measure that physiologically with like skin conductance or the eyes or any of these kinds of responses here. You might wonder what the control conditions are for this. Do people know what the standard one is? So what you do is that you would stroke the dummy and the real hand out of sync. So instead of doing this with the real and the dummy hand, you do this and then this, so that the two are not matched. So what you see and what you feel, and you won't get the same startle response. You won't get the same measurements on the questionnaire, which look like this. So these are the questionnaire responses. The ones underlined are the ones that discriminate the rubber hand illusion more than the other ones. So people say things like, I felt as if the rubber hand were my hand. It seemed as though the touch I felt was caused by the paintbrush touching the rubber hand. And it seemed as if I were feeling the touch of the paintbrush in the location where I saw the hand touch. They tend not to agree with control questions here, so it felt as if my real hand were turning rubbery. It would be interesting if that was the case, but people don't agree with that. So again, this is another way of showing it's genuine. It's not that people agree with any old nonsense that you give them. There's a set of things that they buy into with this. So where does this leave us in terms of thinking about multisensory maps and what the parietal lobes are doing here? In effect, to generate the illusion, we have three different kind of maps of space that are giving conflicting information, and the illusion arises because you're trying to resolve that conflict and figure out what the truth is. So first of all, we've got visual information about where your hand is. But remember, the visual information just tells you there's a hand in front of you. Your vision doesn't tell you whether it's your hand or somebody else. Obviously, somewhere in your head, you know it's a dummy hand, yeah? But your eyes and the information going to your V1 and in and so on is just telling you there's a hand in front of you. That's all your visual information knows. You have then a tactile map of space. So you have your body surface has its own particular map of receptors. But all your tactile map knows is that your hand is being touched. That is all it knows. So at this point, all you know is that there's a hand in front of you being touched, and your hand is being touched. What you then have is another kind of spatial map, and this tells you where the joints of your body are in space. So when I kind of have my arm kind of bent, there are stretch receptors in my joints that in effect tell me what the angle of my various body parts are. And this is called proprioception. It's different from your sense of touch, but it's not a million miles from it. The neurons that are involved in touch are similar to this. It is related. Technically, it tends to be classed as a different kind of sensory information. So in effect, what we've got here is that your proprioception knows that your hand is over there. You've also got a higher order belief that you know your hand's in there. But your visual information is telling you your hand's in front, and your visual and your tactile information are telling you that both are being touched at the same time. And the question is how you resolve this ambiguity between these different sources of spatial information. And the answer is that vision wins. And vision wins a lot when it comes to space, because basically vision is very accurate at locating where things are. Knowing where your limb is in space is not too bad, but it's not as accurate as vision. Knowing where sounds are in space is not as accurate as vision. So there are some situations in which sounds win, but when it comes to space, vision tends to beat all the other senses, unless you have low vision or something like this. So what happens in that is that you kind of integrate this information and you believe that the visual information is true, and you discount the information from your joints that tell you that your hand is over there. So this is what I mean by different maps for different senses. So vision is, at least in early visual cortex, eye-centered. So as you move your eyes around, all the visual information on the eyes moves around, all the information in your visual cortex moves with your eyes. Whereas your body has these kinds of representations. So one is for touch and one is for body movement. This one here is for movement, for action. And again, you can see that the hand has a very large kind of neural representation. You've got lots of neurons that respond to your hand and your face, and less so for the others. That's very receptive to touch and so on. But essentially, in the rubber hand illusion, all you know is that your hand is being touched. You don't know where it is. You just know which body part is being touched in that case. Whereas hearing is head-centered. So basically, the information that comes to you via sound goes in, obviously into your ears, but it depends on where your head is. So as you turn your head, you will get different sounds, even if your eyes are in the same place. And in effect, what the parietal lobes is having to do is it's having to superimpose these different maps of space onto each other in order to figure out whether a sound is coming from over there or over here, and how to link a sound source with a visual source, given where your eyes are. So let me kind of explain that with some more examples. So here, this is our kind of motor cortex here, and this is this strip here. So basically, what this means is that you've got neurons here that respond to the hand, neurons here that respond to the face, neurons here that respond to the tongue. So as well as having neurons that are tuned to different body parts, we also have neurons that are tuned to different directions of movement. So for instance, some neurons, for instance, in the hand area, will fire when the hand moves this direction. Some neurons will fire when the hand goes in that direction. So you've got different neurons that are tuned to different directions of movement and to different parts of the body. And what you're in effect wanting to do when you're trying to move towards an object is that you're having to link the information about where your body is with where information is in a visual centered space. And this is what's called kind of sensory motor transformations, or remapping, or coordinate transformations. And essentially what you're doing is that you're taking a kind of a visual map in which you can imagine the origin being the center of the eyes, and superimposing it on a body map in which the origin is, say, your hand, or the origin is some other part of your body, your head or your trunk, depending on what it is. Okay, so it's like kind of overlaying different maps of space, and that's what your parietal lobes are doing. But it does this across multiple sensors. So it integrates spatial information from hearing and then information from your body around touch and how you want to move your body with your motor cortex with vision information from your dorsal stream here. And this is kind of going into more detail, but as you kind of go into your parietal lobes, you find that there are different kind of subpopulations of neurons that do different things. I won't go into them all, but basically, again, a lot of them focus on this parietal and these kind of frontal regions. And again, these are regions that will be active in the rubber hand illusion, for instance, that are trying to link together where your body is with what you're seeing in the world with regards to your hands. So this region here is called the parietal reach region. And this is the region that, breaking it down earlier, is involved in kind of optic ataxia. So where patients who struggle to post their hand into oriented slots and things like that, that they might struggle to reach towards visually guided objects that are in front of them. So in a way, we could say what this map is doing is it's trying to link together visual information about objects with information about where the hand is. So damage to this region results in this problem of kind of reaching towards objects. The other thing about this is that the neurons respond to the direction of arm movements relative to gaze. So it depends where you're looking at as to where the arm will move. And that's very different from your motor cortex. So your motor cortex will respond, or the neurons would respond to, say, a movement at 30 degrees up there, irrespective of whether your eyes were open or closed, or whether your eyes were looking over to the left or the right. So these neurons here in the more frontal lobes are kind of centered on the body, if you will. Whereas these neurons here are centered on the eyes and guide the body. Okay? Or centered on objects that are relative to the eye gaze. Okay? So although these neurons respond to reaching of the hand, one is in eye-centered coordinates and one is in body-centered coordinates. What's also interesting is that these neurons are specific to the hand. So what this means is that you can look at the right location even if you can't reach for it. So you have an even finer level of dissociation. Whereas here you've got another region called the lateral intraparietal area, which guides the eyes rather than guides the hands. And here this is again in eye-centered coordinates, so it depends, it will encourage you to shift towards the left or the right if you see something, say 30 degrees to the left or the right. But these regions here also, we could think of them as being involved in attention or in kind of selection as well. So for instance, if you take a monkey and you record from these regions, but the task is that the monkey can choose whether to move its eyes or whether to move its hand. So it's a stupid task, there's no right or wrong answer, it's just like a free will. Are you going to look at it or are you going to reach for it? And either way you'll get your juice or your reward or whatever. But what you find here is that neural activity in these regions kind of ramps up before the decision is made to move the eyes or that. So it's as if it's kind of making the decision as to whether or not the reach area goes up or the eye area, LIP, goes up. And we can think of this as kind of analogous to the example of whether you're attending to faces or houses in the ventral stream. But here, it's a question of whether the attention is about an eye movement or about a reaching movement. You've got this ramping up of neural activity that then drives either your hand or your eyes to that. But basically, as you go along, you've got this transition from things being in eye-centered coordinates to things being in body-centered coordinates. So it becomes more abstracted away from the sensory organs the further up the dorsal stream you go. These particular areas that guide eye movements also respond to sounds as well. So this is the area here that is responsible for prioritizing eye movements. So it can generate or leads to the generation of eye movements. It doesn't actually trigger the eye movement, it just plans it, I suppose is the way of saying it, according to the visual information. But what you find here is that, that it also responds to sounds as well as vision. So in this case, what happens is that a person is looking straight ahead, and you can look at where the receptive fields are for sound. So this particular neuron, who knows where it is responding to sounds that are 20 degrees towards the left. So you're looking straight ahead, you play a sound here, the neuron fires. So again, this is like a spatial map here where neurons are tuned to different sounds in space. What's interesting here, though, is that the person moves their eyes over here. And now the neuron still responds 20 degrees to the left. But remember here that the sound is going into the ears, exactly the same thing. So sounds are ear-centered, not eye-centered. So what's happening here is that you are shifting the location of a sound and putting it in eye-centered coordinates. So wherever your eyes go, the sound source moves with your eyes, which is slightly bizarre because it's not moving. Your ears and your head are not moving. Your brain is moving around virtual objects in order to align them and integrate them together here. The other thing about these regions in the parietal lobes is that although they respond to vision, they are very selective in what they respond to. So whereas your visual cortex, so V1, kind of responds to pretty much anything that's in its receptive field, these particular parietal regions are what's called sparse, so they respond to some stimuli more than other. And in particular, they kind of have this property of responding to things that are relevant more than other things. So, for instance, they would respond to stimuli that are unexpected. So, for instance, the classic example is a bang or a flash of light. These will trigger activity in these parietal neurons. But if there is a constant light source or a constant sound source, the neurons don't necessarily fire, except that is if you're attending or if you want to tune in to that particular sound source. So, whereas your sensory cortex responds to sounds and light per se, your parietal cortex tunes into those that are relevant to what you're doing, or potentially relevant if they're kind of alerting you. And this is also kind of how these neurons are kind of coding for attention, is that they are making decisions about what kind of information to respond to, and they are selecting from alternatives. So the idea here is that what's happening is that your parietal lobes are kind of creating salience maps. So salience maps that are centered on the eyes and the hands and so on, that aren't just kind of regular visual maps. They're maps of the important things in visual space, the kind of thing that is relevant to what you want to reach for, the kind of things that are unexpected, and so on. So they're not just duplicating information from below. So what you've got here is information that's kind of flowing through the dorsal streams, that's selecting information according to whether it's relevant to the hands or the eyes, and it's selecting information according to whether it's expected or unexpected. And then your frontal lobes are what's important for actually executing the action, so moving your hands or moving your eyes. As I mentioned before, this is your motor cortex here that contains representations like that. What's interesting is that pretty much all of your body is here, apart from one thing, and that's your eyes. So you do not move your eyes with your motor cortex. They have their own separate driver, if you will. And these are what's called the frontal eye fields here. So if you were to apply electrical currents to this part of the brain, people will start to make saccades. They move their eyes around, whereas if you apply electrical currents there, people will move their face and their hands and whatever. So the eyes have their own separate controller in the frontal lobes called the frontal eye fields. And essentially these two regions here, the parietal lobes, which is kind of selecting visual information, and your frontal eye fields, which are generating saccades, are kind of part of your attentional circuit for orienting to visual information and also non-visual information that is relevant to eye movement. So although these frontal eye fields are involved in triggering eye movements, they also feed down from the top down into the parietal regions as well. So as well as being the end point of action, they also, the information goes backwards and forwards. And essentially we can study that by stimulating this part of the brain and seeing what happens to visual attention. So if we stimulate neurons in the frontal eye field strongly, what happens is that you will move your eyes. And again, you will move your eyes in a particular thing. So some neurons will code for an eye movement here, some neurons to an eye movement there. So we can kind of map individual neurons where they trigger eye movements. What happens if you stimulate these neurons weakly so it doesn't trigger an eye movement? What happens then is that basically you are good at seeing information in that particular part of space that your eyes would have moved to had they triggered that information. So in effect, if you stimulate the neuron that moves my eyes to here weakly, I will be good at detecting visual information there. Now what this means is that that's kind of a neural basis of looking out of the corner of your eyes. In effect, what you're doing is that it's a non-executed eye movement. You're saying, I want to move my eyes there, but I'm not going to do it. And that is enough to kind of amplify the visual stimuli there. So basically what this graph shows here is your ability to detect visual stimuli in the periphery in one of these locations that are being stimulated. So luminance change there means how bright it has to be. So what it means is that when this non-executed eye movement is stimulated, you are good at detecting faint stimuli here. If you don't stimulate that part of the frontal eye fields, you need a higher brightness in order to see it. So in this way, action isn't just kind of the end point of cognition. It's not just what you do when you've found your visual stimulus. Action actually kind of drives your ability to see things that are out there. And we can think of looking out the corner of our eye as literally an eye movement that has been planned but hasn't happened. So as I stare here and I move my attention around, it's triggering eye movements, but I'm not executing them. But it's helping me to detect information in those parts of the world. Okay, we'll have a break for 10 minutes and then we'll talk about visuospatial neglect. Okay, we can make a start. So basically, to kind of recap where we are with this, that basically when we process sensory information, it goes down multiple routes. We talked about lots of different kind of subroutes. Last week, primarily going down the ventral stream. And today we're talking about the dorsal stream. So the ventral stream is important for knowing what objects are, whereas the dorsal stream is important for knowing where they are. So they have these kind of spatial receptive fields that they respond to where things are in space, less so than what. But they also respond where things are in space in different kind of what we call maps or coordinate frames. So some that are eye-centered, some that are head-centered, some that are hand-centered, and so on. And they respond to locations not just from vision, but to the body, to hearing, and other senses, so we can pull that kind of information together to figure out where things are in space and to be able to act on them. This is also important for attention as well as action, because it acts as a kind of a filter of sensory information. Although it responds to sounds and vision, it does not respond to all sounds and vision. It responds to those sounds and visions that are more important, either because they're unexpected or because they're what you are searching for in your environment. So looking for green lines or whatever it is, you will activate those within your parietal lobes more than your visual cortex. So that's the kind of the essence as to how this works without going into details. What I'll talk about for the last bit is the difference between the left and the right hemispheres, particularly the parietal lobes, in spatial attention. and neglect. So first of all, the parietal lobes work rather differently from the early visual cortex. So remember, in the early visual cortex, the left hemisphere responds to the right side of space only, and the right hemisphere of my early visual cortex responds to the left side of space. Your parietal lobes also have a hemispheric specialization, but it's not as drastic as your visual cortex. And what that means is that my right parietal lobes will respond to the left side of my right parietal lobes will respond to the left side of space very strongly. It'll respond to the midline a little bit, and it'll respond weakly to that side. So your parietal lobes contain information about all sides of space, but it contains strong information about the opposite side of space and weak with the other. Whereas in the early visual cortex, you've got a sharp cutoff between left and right. Here, it's a graded response that you have. And your left hemisphere does the opposite. So they have these rather complementary profiles here that they are tuned to the opposite side of space. Your left hemisphere responds more to the right side of space and the right to the other. One of the things that you also have is that although they have these kind of opposite tuning profiles, that one hemisphere is more important for spatial attention than the other. And this appears only really in humans. You don't see it in other primates. But the idea here is that the right hemisphere is more specialized for spatial information than the left. It's not entirely clear why. I mean, the best guess is that it's nothing to do with spatial information. It's to do with language being in the left side, and this is the compromise that you have, rather than needing space to be represented in this particular way. What this means, though, is that although your left and your right hemisphere are capable of orienting space, the fact that you've got more spatial representations in your right hemisphere means that people naturally are more aware of things on their left than they are their right. And this does seem to be the case, and it's called pseudo neglect. I don't know how well this works, but the idea here is that you have to say which one of these looks darker. And I kind of have a sense that I want to say that this one looks darker than that. And of course, you know that they're mirror images of them, so the amount of luminance information from this bar and this bar is the same. But there is something about the left side of this, and it seems to be relatively cross-cultural as well. If you look at kind of accidents in A&E, people bump their left, hang on, which way around? People bump their right side more than their left side. On average, that they are less attentive to one side of their body than the other. And you see it kind of in art and so on, that you're more likely to read an image from the left to the right and so on, or have certain spatial motifs like the sun will be in particular locations more on one than the other. Yeah.
Sorry, maybe I just missed it. Did you say that people injure their left side more often than their right side?
They are more attentive to their left side, so they will be injuring their right side more. Yeah, exactly, yeah. It has to be that way around. And this is just kind of helping people. You'll notice this perhaps if you go on a walk. So one of the things that I've noticed is that we go on a walk that has a sheer drop on one side, but it's far worse when you're walking with a sheer drop on your left side than the right side. And it's nothing to do with, well, obviously partly it's depending on your eyes, but assuming you're looking straight ahead, that will be the sensation that you have of this. So look out for it. It's supposedly not related to handedness. There's somebody called Mike Corballis, which has looked into this as well, a guy in New Zealand. It's not strongly related to that. It might be related to where language is in your brain, which is a different thing from which hand you use.
Sorry, you just mentioned it might be related to languages. You mentioned that this is a cross-cultural phenomenon.
It is, yes, that's his claim, yes. So related to language in the sense of whether language is in your left hemisphere or your right hemisphere, rather than whether your language reads left to right. But that's the case here, yeah. Yes, if you're somebody with language in your right hemisphere, you might have the opposite profile, but it doesn't matter whether you're Chinese or English or Arabic or whatever. Yeah. So they say. So basically, in people without brain damage, what this means is that we're over-attentive to the left, what this means in people with brain damage is that damaging one hemisphere can have particularly more drastic consequences for attention than the other. And obviously, if you damage your left parietal lobes, then basically you will still be able to deploy attention to both sides of space. You will have, in this case, you will be over-attentive to the opposite side of space here. But if you have damage to your right hemisphere, basically this has far more drastic consequences because the right hemisphere is more specialized. It contains far more neurons coding this. And in effect, what we could imagine is that both the left and the right kind of parietal lobes are kind of coding what we'd call a salience map. So they're deciding what kind of information in the environment is more important. And normally it will prioritize information on the left. But as you damage one hemisphere, then the other side becomes more important. So I'll talk about what the characteristics of this is and what it tells us about how attention and space is kind of organized. Yeah, so these are the kinds of clinical tests that you would give to patients with neglect. So one is, as in the video here, is to copy a drawing and see how many features are missed. And typically a patient will miss the left side because, remember, the right parietal lobe causes this damage. You can get neglect on the other side, but it tends to be a lot more mild because of this hemispheric asymmetry in how attention is deployed. If you ask people to draw from memory, again, they might miss objects, but they will remember that there are 12 numbers on, they will go and write them on, but somehow they've spatially misorganized them. Here, what they're asked to do is just put a line through all the other lines. So they've got a blue pen, and they're just asked to cross all the lines. But again, they don't necessarily cross the lines here. The further away it is on the bad side, the less likely they are to do it. And here, this is line bisection, where the task is to put a blue line through the middle of a line. And if you look at the long line here, it's as if they've kind of neglected this part of it here. So the true center of that line would be about here, okay? But they have, they're kind of not deploying attention to that, or they're, some people say that it's like having space kind of contracted on one side. I'm not sure whether that's the right analogy. It is caused by damage to the right parietal lobes. Again, you can have it with damage to the left parietal lobes, but the symptom would be weaker, and often the patient would be able to compensate for it quite well. But again, there's evidence that neglect isn't just one thing, that it's multiple things, and this kind of corresponds to our idea of there being multiple spatial maps. So in particular, it might be often the case that it's the spatial map that's involved in kind of triggering eye movements, which is why the patient doesn't even think to look over there, is because there's nothing to tell her that there's something interesting to move her eyes towards, for instance. But it can be body-centered, and I'll talk about this. So here, because this looks like a disorder of vision, I just want to kind of labour why it's a disorder of attention and not vision, even though it's affecting the processing of visual information. So if you were to do fMRI of somebody with damage to their right parietal lobes, they will activate their occipital lobes and their ventral stream, even for objects that they say that they're not aware of. So here it might be the side of the cat, or you present a left and a right object simultaneously, and they will, let me get it the right way around, they will ignore the left object and report the right one, but you could show that that is still being activated in the ventral visual stream. And also that they can often detect objects if they're cued there. So again, if there's something unexpected, or the expert says, go and have a look at that, then they're quite surprised to think that they missed it in the first instance. But remember, although this sounds very odd, that we often miss things that are in plain sight from us. Also, it isn't just a visual phenomenon. It affects auditory and tactile judgments as well. So it's not necessarily the case that a sound on one side is, sometimes they're missed, but often they can be mislocalized, that they appear more central than leftwards than they are, for instance. Or that if you touch them on both the left and right side of their body, often they will not notice the thing on their bad side of the body. This is a good example of what it means of why it is an attention rather than a visual deficit. So here, what you do is you present these kinds of stimuli briefly, so maybe 200 milliseconds, and the person is looking at the center, and the reason you do this is that so they don't move their eyes, and you ask them what they see. When you present them here, they say, I see a frog, and when you present them here, they say, I see a sun. Now this is puzzling because here they've got stimuli on both the left or the right side. What's interesting is what happens when they see two stimuli, one on the left and one on the right. And here what they say is, I see a sun. So even though they can see a frog under normal situations, when there are multiple competing stimuli, and remember that attention is all around kind of competition, that one stimulus wins the competition, and that's the one in the correct, or the kind of intact, or the better visual field. But this kind of makes sense if you think about the inattentional blindness with the gorilla, that you only don't see the gorilla because there are competing stimuli in your visual field that you are doing. It would be trivially easy to see the gorilla if it wasn't. And this is the same thing here. It's trivially easy to see the frog when there isn't competing stimuli in the visual field. But this suggests that it is to do with attention and kind of competition amongst different kind of visual representations rather than seeing itself. This is the other kind of evidence. So this is a study by Wayumia. And basically what they would do is that they would present this particular image in the neglected field. And they ask the patient, what did you see? And they would say, I didn't see anything. Then what you do is that you present them with these kinds of stimuli and you ask, what is that? Now basically, if you were to present these stimuli without having seen that, people would require a lot of information. Maybe they have to see this to figure out it's an apple. Whereas if they've already seen that before, they might get it here with less information. And this is the phenomenon of perceptual priming, in effect, that you can use a kind of recent perceptual memory in order to boost your ability to see. But in order to have a perceptual memory, you must have been able to see that at some level in the first place. So even though they deny seeing it, they can do that. The burning house experiment is that basically you've got two identical houses, and one house has flames coming out of the left side of it. And you ask the patient, can you see a difference between the house? And maybe they don't, particularly if you present it briefly enough. But then you ask them, which house would you like to live in? And they point to the one without the flames. Okay. So again, they have some instinctive idea that there's something not good about that house. So this suggests that they are kind of perceiving information, in this case at the level of some kind of semantics, or certainly holistically rather than this, and here that they're getting some emotional information about stimuli that they are not attending to. What we also have is information between different kinds of spatial map. So here this was looking at attending to external information versus internal information. So looking for external information would be these kinds of tasks that I've just shown you here, where you've got to kind of find objects on a page, draw objects, or here when you're doing that. What is kind of internal imagery? Well, this is things like imagining your way around a familiar location. And here they did it in this square in Milan, but you could imagine doing it in Churchill Square in Brighton or Trafalgar Square, where you imagine, say, standing on Nelson's column and looking at a particular way, and you have to describe all the buildings around you. What you find is that some patients with neglect only describe the buildings on their good side. This would be stimuli on the right side. If you've got damage to the right hemisphere, you can't attend to the left side, so they only describe one side. What you can then get them to do is to orient to the opposite end of the square and describe it, and now they report all the things that they missed last time around. So it's not that they've lost their memory for the scene. It's just that they are only attending to objects within that particular attentional frame of reference. And as you shift your perspective, you tune into different features. What's the interesting thing about this is that some patients can do the task that they only have neglect for imagined space, but not for real space. And some patients have neglect for real space, but not imagined space, that they can do this task. So again, it suggests that there are either different spatial maps or different routes for accessing these maps that have different kind of neural components within the parietal lobes. And again, because these are coming from patients, we don't necessarily have a good idea of where they are, but yeah.
If you show a patient a picture of a snake or something that we're programmed to be scared of in their bad side, would they still be able to be afraid of it?
I think that supposedly the answer is yes, that they can, yeah. If you flash it briefly, then they might not report seeing it, but they might have something like a skin conductance response, yeah. I think that that's the idea behind it. You can also have a dissociation between near and far space. And again, this has been motivated by evidence from neurons in the monkey parietal cortex, that some neurons will respond to visual information in near space, which is flashes of light, where the end of your fingertips is, and other neurons respond to flashes of light outside of that. So even though they're in the same retinal position, you have neurons of the parietal lobes that respond to depth. And this is important because obviously if you're reaching for objects, you need to know where they are relative to your body and whether they're within your reach or outside of your reach. And there is a very precise definition of near and far, and it's where your fingertips end. Because at that point, in order to reach, you've got to move your body. You cannot move your hands. You've got to move your whole body. What you find is that you can also have patients who have neglect of near and far space. So some patients struggle to find objects, say, on their left side of near space, for instance, on paper and pen tasks, but they can find objects on the left side of far space and some the other way around. What's also interesting is that what's defined as near and far is kind of the end of your fingertips. But if you're using a tool like a ruler, then near space gets extended out by, say, 30 centimeters. And you can also show this in the single cell recordings of monkeys as well. Your kind of bubble as to where your body ends becomes extended by holding tools in your hand. And that's the same for a monkey recording from a single neuron in its parietal cortex. And it's the same for a brain damaged patient who, for instance, has problems in far space. If they're holding a tool, then all of a sudden near space becomes far space and their deficits start to kind of ameliorate. So you've got these very interesting parallels between the level of kind of single neurons in a monkey and these kind of neurological symptoms that you see here in some of these patients. But again, the idea here is that you're kind of embodying objects, that the objects become part of you, in effect. So we talked about kind of body-centered kind of maps of space and the kind of more external maps of space, whether they're coded relative to eye gaze, for instance. But again, this distinction also plays out in neglect. So there are some patients who neglect half of their body but don't neglect things visually. So to neglect half of your body means that, for instance, that you only put makeup on one half of your face or only shave half of your face, but you might have no problems in dealing with information externally. So the non-bodily equivalent would be having a plate of food and missing the food on the left side, for instance. So you can have patients who don't groom half of their face but then get the whole of the plate, and some patients who miss food on half the plate but fail to do that. And again, the idea here is that you have multiple different kinds of neural representations of space, and neglect can impact on how you access or prioritize information in those different maps selectively, depending on the nature of your brain damage. The other one that we kind of have is what's called object-centered and kind of object-based neglect as opposed to kind of more space-based. So have a look at these, what these patients here are doing. So these neglect patients have been asked to circle all the letter A's. Now what you could imagine that the patient would do is that the patient might circle all the letter A's in this column but miss the ones in there. Whereas in fact what the patient is doing is circle all the letter A's on the right side of this column and the right side of the column but miss the ones in between. So it's almost as if they put an attentional focus here, then move the attentional focus to there. So the problems within their attentional map is kind of within a set of objects or within a particular spotlight, but as the spotlight moves, it's kind of intact. Or similarly here, you can ask a patient, for instance, to draw this scene. Now, some patients with neglect would draw only the objects on the right and miss the ones on the left. Okay? Whereas other patients draw every single object on the scene, but just miss one side of each object. Okay? And here, again, you can imagine that your kind of attentional spotlight or your kind of map here is either just centered on the scene as a whole, or it's centered on individual objects, and you move the spotlight to each object and carry on missing one side of every single object as you do so. What you can also have here is that this is another example of object-sided neglect, where basically you just have to say whether these two objects are the same or different. Here they're different, and the thing that tells them apart is this little kind of knob sticking out here, this little square. What's interesting is that if you put the objects on one side, so here the bit that you have to detect is on your bad side. The left is typically bad for neglect patients because they've got damage to the right parietal lobes. But if you rotate the object, now the difference is on their good side of space, but on the bad side of the object, assuming that the object has its axis oriented like that within it. So patients with object-centered neglect are bad at both this version of the task and that version of the task. Whereas patients who are just neglecting space defined in terms of the midline would have differing performance depending on how you're defining that here. Whether you're in object-centered coordinates or kind of more space-centered coordinates, what's a better way of describing it.